This is Seed Germination, From Seed to Seedling by Trever Wentland, Ahmad Jamil, and Hassaan Qureshi. Today we'll begin talking about the story of a simple seed can make the transition to a plant. And how does the process begin, you might ask? By germination. What is germination? Germination is the process in which a plant grows from a seed. Specifically, it is a process that occurs from the end of seed dormancy until shortly after the protrusion of the radicle. This might sound quite simple, but indeed it's quite not, as there's many factors regulated from physical to hormonal. Yet this process is quite obviously important, as without it, seeds would never be able to leave their coats and grow. It's important to remember, though, that seeds, most of them, start out in a state of dormancy. Dormancy is used as a way to keep the seeds from germinating, and as such, the seed is kept in a reduced state. So before the germination can begin, the seed must leave the state of dormancy. To leave dormancy, the seed must meet certain requirements which are unique to each seed. Although one thing is somewhat constant between all dormancy releases, that being the concentrations of abscisic acid and gibberellin is paramount to dormancy release. So what are the examples of requirements germination might need? Well, first, the seed needs water, as the dormant seeds are nearly completely dehydrated, so the water is necessary for the activities of the seed, such as providing oxygen to the seed, softening the seed coat, and increasing seed permeability. Oxygen, as mentioned, is important as well, as it plays key roles in aerobic respiration, a process necessary for the creation of ATP for plant cell function. The seed also needs to be kept at viable temperatures and pH if the seed is to stay functional during development. Lastly, many seeds have special requirements for lighting conditions. Unlike you might think, not all of them require high light. Some seeds have some especially interesting things that may be required for germination to begin, such as being subjected to intense heat, or after intense cold, such as a snowstorm. Some require being digested, some require being washed to help remove inhibitors on the outside of the seed, some may require being damaged in a process called scarification, in which the seed coat is torn. And that brings us to our first clicker. So, why might being digested help promote seed germination? A, B, or C? Well, the correct answer is A. It would help to erode the seed coat so the seed would be able to sprout. This is important because it exemplifies the seed coat, an outer coating that helps to protect the seed, and it also becomes a nuisance when the seed's trying to get its first roots out of the cell, a step many people overlook. Talking about leaving the seed, there are three ways in which our seed might germinate. In the first, hypogeal germination, the cotyledons do not come out of the soil surface, but instead the epicotyle elongates and pushes the plumule out of the soil. If our little seed is a monocotyledon, then it utilizes this style of germination. In the second, apical germination, the cotyledons are brought above the soil via elongation, where the cotyledons help perform photosynthesis till the seedling is capable of doing it on their own. Lastly, there's a vaporous germination, and unlike all the others, it lacks seed dormancy. With embryos growing right off the mother plant and having the radicle elongate until it becomes so heavy it falls off and forms the first roots in the ground. So now we know the types. How does germination begin? Well, long before the radicle can emerge from the seed coat, the newly active seed must first uptake water. This process is called imbibition, and it has two phases. In the first phase, right after leaving dormancy, the seed rapidly uptakes water, leading to the seed swelling immensely. This swelling ends up deforming the membrane structure temporarily and beginning protein synthesis of existing mRNA. After the seed reaches a point of stable water content, the water uptake slows and is in phase 2 now, in which the DNA and mitochondria are pairing and growing in size. The embryo now begins to expand, with the seed covering, called the testa, being weakened and broken down. And once the seed is properly hydrated, the seed coat has been reduced enough, the seed utilizes large amounts of energy to rupture the testa. This rupturing activates the root apical meristem and starts elongation of the radicle, which grows through the tear in the testa, forming the first fruits in the seedling and concluding germination. While the germination process of the seed is ended, the understanding of it has just begun. As previously mentioned, this process is heavily regulated with hormones, so some of which you might have even heard of before. So please stay tuned and hear from our next speaker, Ahmad.
Two fundamental hormones that play a crucial role in seed germination are abscisic acid, abbreviated as ABA, and gibberellins, abbreviated as GA. These two hormones are coined as antagonistic hormones due to the fact that they result in polar opposite effects for seed germination. Abscisic acid establishes and maintains seed dormancy, whereas the gibberellins promote seed germination. The balance between ABA and GA is regulated by environmental cues such as light and temperature. It's the disruption of this delicate equilibrium between the two hormones that causes seed germination to be promoted. Specifically, when we see an increase in GA or a decrease of ABA, that we see signs of germination. Understanding the metabolic pathways for these hormones can help us have a better understanding of how these hormones are regulated in the seed. The abscisic acid metabolism is one that is complex and functions to ensure dormancy with the production of ABA. The process in which ABA is produced consists of a series of enzymes and an integral oxidative cleavage process that breaks down and alters several intermediate compounds. When we finally produce ABA, the seed can choose to hydroxylize ABA to inactive forms called hydroxy-ABA. This would cause ABA concentration to drop, which can potentially give way for germination to occur. Therefore, the balance between the synthesis and catabolism of ABA in the metabolic pathway can cause changes to the overall ABA concentration in our seedling. Now we're going to talk about the other hormone, gibberellin. This is the most important hormone for seed germination to occur. A high level of gibberellins is required for the counteraction of ABA activity in the seed. The spike in GA levels activates responsive genes which induce cell wall remodeling enzymes. This leads to the weakening of the embryo surrounding area. One important thing to note is that there are over 136 natural occurring GAs, but there are only a few that are considered bioactive. This consists of GA1, GA3, and GA7. One thing that you'll notice about the gibberellin metabolism chart is that there are many forms of GA. The important GAs in this metabolic pathway that will actually have an effect on the plant are the ones circled in red, GA1 and GA4. The rest of the GAs are precursor catabolic products. The production of these bioactive GA1 and GA4 can be repressed by the enzyme GA2-OX, which converts the bioactive forms to inactive forms of gibberellins. Ultimately, the regulation of the synthesis and inactivation of these bioactive GAs can allow germination to occur. So when our little seedling sees an increase of GA1 and GA4, there will be a higher chance of germination to occur, considering all the other necessary stimuli, such as light and temperature, are present.
The first notable growth regulatory hormone that affects seed germination and dormancy is auxin. Auxin provides a signaling pathway involving a cascade mediated by transcription factors. The amount of auxin present can shift the precise balance of ABA and GA, determining whether the plant will go through germination or dormancy. In general, having high auxin levels will promote seed dormancy. Auxin uses the response factors of ARF10 and ARF16 to induce the expression of ABA for seed dormancy. The role of auxin for germination has been merely studied, so not much is yet to be found about its role in germination. However, studies show in reversing ARF10 and ARF16, seed germination is promoted and precocious germination rates are also higher than the wild type. A balance of ethylene must be kept to find the common middle ground and regulate crosstalk of ABA and GA that best allows for germination. ETR1 and EIN2 mutants show an increased level of seed dormancy in relation to the increased level of ABA content. SNL1 and SNL2 mutants at the epigenetic level reduce the acetylation level, ultimately leading to the promotion of seed dormancy as well. In addition to ethylene, its precursor ACC also provides a less effective form of seed germination. Jasminates are hormones that regulate a plant's adaptations to abiotic stress. The effects of jasminates are minimal on seed dormancy. However, specific examples have rose over time. The accumulation of the jasmonic acid precursor causes increased dormancy only in a comatose mutant. Adding exogenous jasmonic acid to mature seeds has also been found to inhibit germination in multiple species. On the contrast to this, adding endogenous jasmonic acid promotes germination. Brassinosteroids are a group of hormones that, of course, promote germination like gibberellins and counteract the effects of abscisic acid. The BR signal is necessary to overcome inhibition of ABA by actively increasing the growth potential of the embryo. BR controls the key genes GPA1 and GCR1. BR also regulates proper seedling growth under stress such as elongation and germination. BR alters antioxidant enzymes, which leads to nutrient accumulation and advanced seed germination. Cytokinins play an effective role in allowing a bud to leave plant dormancy by creating a buildup of growth promoters and a decrease in the amount of inhibitors. An example of a cytokinin is kinetin, which synthesizes the light-requiring seeds so that the plant germinates with a lower intensity of light than regularly required. The greatest number of seedlings was found in the cytokinin thidiazuron. In conclusion, it is found that plants placed in a cytokinin media and then removed and placed in a normal medium will then produce a larger number of seedlings. Pause here and answer this question, please. Abscisic acid is the only of the three that's a positive regulator due to its ability to allow the seed to exit dormancy and be primed for germination process of dormancy release. Breaking dormancy in ideal conditions will lead to the uptake of water, increasing metabolic activity and inhibition. The water uptakes will become stable and through where the seed covering will be weakened. This allows for the first radical to protrude from the cell and ultimately establish the connection between soil and plants called roots.